Okay, welcome back to the Classics in Immunology Journal Club. Before we get started, don't forget on the YouTube channel to like the video and subscribe. Click on the bell to get notified. Scroll down and check out my website. And on the website, check on the writing tab, and you'll find all of the classic papers, including the one we're going to talk about today. Today, we're doing another one of my favorite papers because it's one of my papers. The authors are Zmuidzinas, Harvey Mamon, Thomas Roberts, and Kendall Smith. And the title of this paper is the Interleukin-2-triggered Raf-1 expression, phosphorylation, and associated kinase activity increased through G1 and S in anti-CD3 stimulated primary human T cells. Quite a mouthful. And I'll tell you why we put all that into the title. This was published in the Journal of Molecular and Cellular Biology in May of 1991. That's also important, as usual, in these scientific circles. Now, the background on this paper is that, first of all, Raf-1 is a proto-oncogene, CRAF1. The people interested in oncogenesis and oncogenes and so forth primarily had used cell cultures, long-term cell lines, to study the expression of these oncogenes. And when you do that, the problem is that you can't really study their how they might function within the cell cycle, stimulating cellular proliferation, because those cells are always proliferating. They're asynchronously proliferating within the cell culture. Some cells in early G1 and some cells have just completed mitosis and so forth. And they're sort of moving along and proliferating like all cells would like to do. And the difficulty is that you can't stop these cells from proliferating. You can remove them from whatever their culture conditions is to sort of starve them of nutrients and in particular serum, and they will sort of wind down and stop proliferating, but they're always really so-called in the cell cycle. And therefore, to try to study the function of different genes and how they might be activating different pathways and so forth within the cell cycle is problematic. In this instance, Zmuidzinas was a graduate student at Dartmouth in biochemistry, and she entered my lab after she'd been in somebody else's lab studying Raf-1 as a grad student. And her family and she descended from people that came from Lithuania. That's why she has an unusual name like that. And she was a very good graduate student. And when we wanted to get involved in the so-called signal transduction business, I asked my good old friend Alice Reinhardt at the Dana-Farber Cancer Institute in Boston to, who's good in the business down at your place, and he recommended that we get in touch with Tom Roberts, who was a kinase guru at the time. In his lab, he had a postdoc by the name of Harvey Mamon, who was an MD type, and he went on to become a radiation oncologist, I've learned. And so Nina and I drove down to Boston to talk over these different kinds of experiments that we might do together, and we arrived on a collaborative effort so that Nina would prepare the cells and keep part of the different preparations back at Dartmouth, and we'd send samples down to Boston for Harvey to do the experiments down in their kinase lab. Julie Stern, I think, recall I reviewed one of her papers that she was in the lab from 1986, 5 years previously, where she had initiated experiments to look at the expression of these different oncogenes in the IL-2 T cell system that Doreen Cantrell had worked out just a couple of years previously, 83, 84, where we could show that we could start and stop the cells, the human peripheral blood T cells, derived T cells. And we know that the nice thing about T cells versus all these other cell cultures, and most of the people in the oncogene business were studying mouse embryo fibroblasts that were called 3T3 cells, and which were grossly abnormal. They had abnormal numbers of chromosomes. They had chromosome breaks, translocations, all kinds of things. But this was the normal, so-called quote unquote normal cell cultures that the oncogene people had been using for their experiments, which I thought was not going to work. The nice thing about T cells, human T cells, is they circulate in the blood so that they're easily obtainable. But in addition, and more important, they are physiologically and biochemically and molecularly in a state of G0, which was unattainable in the mouse embryo fibroblast system. You had to, in order to study them growing, you had to add stimulants to them in order to get them growing. We had already shown, and I think we've covered it sort of ad nauseum here, but this time in the early 90s, we were able to activate these cells via their T cell receptor signal transduction complex, the so-called CD3 molecules, and we had different antibodies reacted with the CD3. And that would initiate a cell cycle entrance into the cell cycle from G0 into G1. Couldn't do that in the mouse embryo fibroblast system. So that was the intro. That was the background. The intro is that VRAF was a virally identified oncogene that was picked up in the transformation system started by Jeff Cooper and Weinberg from MIT. And VRAF was originated in a Maloney sarcoma virus, mirroring Maloney sarcoma virus. It was first published in 1983. So this is not even a decade later. And CRAF, the cellular proto-oncogene, was identified in 1986. And so at that time, Julie was in the throes of doing her experiments, and VRAF was one of the genes that she looked at as to see whether or not it was activated either by the T cell receptor or by the IL-2 receptor. And some interesting data came out of her studies, but they obviously needed to be followed up. And so Julie worked on CMIB from the myeloblastosis virus. And so RAF seemed like a pretty good candidate. And Nina had already had experience with RAF at another lab at Dartmouth. And so it was, as the saying goes, the rest was history. Now, it turns out that by this time, RAF had been pretty well characterized at the molecular level. It encoded the gene encoded a 70 kD protein that had two domains, a 5' regulatory domain, which was a negative regulatory domain, and then a 3' kinase domain. And so if you mutated the regulatory domain, it would allow the activation of the kinase. And that's how the VRAF essentially lacked the whole regulatory domain, and it was just the kinase domain. And then they did other kinds of mutation studies to figure out what was going on. They found that the active VRAF and also 5' deleted CRAF would put onto the mouse embryo fibroblast cells would cause them to proliferate. So therefore, they decided they concluded that it must signal cell cycle progression. And so the question was, how does it do that? And that's what we were approaching in this particular paper. They'd also shown by this time that the RAF protein in fibroblasts would associate with the platelet-derived growth factor receptor. It would actually bind to one another, those two proteins in the cell, and that would activate the kinase activity. The PDGF receptor had been shown or thought to activate G0 to G1 transition, the same thing that the T cell receptor does. The nice thing about the IL-2 system, as I just mentioned, is the fact that you could separate the T cell receptor signals from the IL-2 receptor signals. And that was critical. You needed to be able to do that. And you could do that easily because you could take freshly isolated T cells and activate them through the T cell receptor and then study them very rapidly thereafter. And or you could then grow T cells in IL-2 for a week or two after they'd been activated by CD3. And then you could take them out of the anti-CD3 and the IL-2 and just let them rest in the incubator for a day or two. And they would all reaccumulate then now in early G1. And then you could add IL-2, and they would then undergo cell cycle progression after that. So the cell cycle people would talk about competence on the one hand, and that's what in the T cell system the T cell receptor did, and then progression, cell cycle progression, which is what IL-2 did. And we had shown that, and that's what Doreen had shown back in 83, 84. That's the intro. And so the materials and methods in this paper, the reagents that we had available by now was cyclosporin A, which we knew blocked T cell receptor activation. We had 4-ball-myristic acetate and 4-ball-dibutyrate, both of which activated protein kinase C and activated T cells via a portion of the T cell receptor system. We had cyclohexamide to block protein synthesis. We had interleukin-2 purified by now and homogeneous. And we had a very special anti-CD3 molecule that was called 64.1 that I talked about in a couple of the previous journal clubs that would activate purified T cells that had been all the accessory cells removed, all the macrophages removed. And this 64.1 anti-CD3 would activate those T cells via the T cell receptor signaling molecules, CD3, without the accessory cells. And that was important because they had a much cleaner system to study these kinds of things than other people had used. In terms of the cells, then we had peripheral blood mononuclear cells that isolated over Ficol high peg density gradients. We had T cells that had been grown for a couple of weeks in IL-2. We had our IL-2 binding assay so that we could characterize the assay for the number of receptors on the cells and so forth. We used from Stefan Ehler's experiments this lysosomal tropic agent called leucine methyl ester that would be taken up by phagocytic cells, and then it would lyse the cells. So you can get rid of the phagocytic cells. And that's one of the ways we cleaned up the T cell compartment. And so that was it. And then to analyze our RNA, we looked at total cellular RNA that was extracted from the cells at different stages by guanidine hydrochloride and cesium chloride gradients. We could then take our purified cellular RNA and then assay it for messenger RNA expression by running it out on gels and then probing those gels with radiolabeled cDNA probes for RAF and IL-2 and so forth and so on. We also did Western blots looking for the RAF protein. We could lyse the cells in a detergent and then run the protein from the cells out on gels and transfer them to nitrocellulose, and then use anti-sera. We got antibodies through the RAF gurus that had purified RAF and then raised anti-sera to RAF. We could quantify the proteins on the gels by densitometry. So we had good systems to at least qualitatively identify RAF, and we could tell whether there was a little bit or a lot on those gels. And we also had kinase assays so that we could take those cell lysates and then precipitate the RAF proteins with anti-sera complex to separose beads. And then we would add radiolabeled ATP and just assay, see whether or not they would phosphorylate themselves. That would be an autokinase assay for other proteins. So let's go to the results and the screen sharing. Let's look at figure 1 from this paper, which is a composite of RAF messenger RNA and protein expression after anti-CD3 treatment. So that's in the A panel here. And we're looking at CRAF. These are northern blots, so all these are. So this is messenger RNA at different intervals after the anti-CD3 treatment. And you can see that it's present at time 0. And then at 3 hours and so forth, it is increased, but it really gets moving around 24, 36 hours. By comparison, IL-2 after anti-CD3 treatment is only transiently expressed maximum at 6 hours in this experiment with the IL-2 receptor alpha chain, P55, is delayed until around 6 hours, same as IL-2, and then peaks at around 24 to 36 hours. And the important thing about these data is that the expression of the alpha chain IL-2 receptor is sort of coincident with the expression of the messenger RNA for Raf-1 increase in that messenger RNA. And here down here in B is arbitrary units and densitometric scans of these kinds of gels to look at Raf-1 message hours after anti-CD3 treatment. So you can see this is why from 12 to 36 hours is where we see peak levels, and they peak out at about a 6 to 8-fold increase during these time intervals. Then we can also look at the mobility shift of that occurs in the RAF protein that's due to phosphorylation of RAF after the anti-CD3 treatment. And you can see what happens. This is that, first of all, at time 0, there's little protein. And then you can see a little bit more after 6 hours. And then after 12, 24, 36, 48 hours, a lot more of the protein. And this is the phosphorylation. When it's phosphorylated, it retards its flow on the gel. So this is the protein itself down here. And then this is the phosphorylated version of the protein. Then in panel E here is the fact that the RAF protein increase that occurs here precedes the onset of tritiated thymidine incorporation. So in the solid symbols, we have Raf-1 protein levels after anti-CD3 treatment. And then this is the thymidine incorporation of the proliferation of the cells. And you can see that the expression of the RAF protein, this is at 24 hours, is clearly discernible at 24 hours, whereas there's no thymidine incorporation. Then at, say, 36 hours, you start to see thymidine incorporation peaking right at 48 hours. And then at 72 hours, we remove the anti-CD3 and the IL-2, put the cells back into culture. And the thymidine incorporation, which is in the open symbols, then falls off over the next few days. So the problem, so that this shows what happens when you activate the cells and then follow them in culture over time and look at this particular oncogene expression, both at the messenger RNA level, at the protein level, and at the phosphorylated level of that protein. It's difficult to sort it all out as to whether or not the T cell receptor is doing all this, or whether the IL-2 receptor is doing all this. So then if we look at figure 2 in this paper, here we've let the cells grow in IL-2 for about 2 weeks, then we've taken them out of IL-2 for 48 hours. And we're looking at messenger RNA in the triangles with IL-2, added IL-2 back again now in these cultures, or not added IL-2. So if you don't add any more IL-2, the messenger is basically baseline. Whereas if you stimulate them or give them back IL-2, within hours, right after IL-2 treatment, you see increased messenger RNA levels for RAF. And that is then followed by tritiated thymidine incorporation in those cultures. So the conclusion from these kinds of data is that the increase in messenger RNA for RAF increases progressively through G1 and peaks before the onset of S phase. And if you don't add any IL-2, you don't get that at all, and nothing happens. Then if we go on to figure 3, we see the effects of anti-CD3, IL-2, dibutyryl cyclic AMP or PMA on the RAF protein mobility in the gel. And just to refresh, when the RAF is phosphorylated, it is retarded in the gel. So after anti-CD3, Here's time zero. We're starting. We're adding anti-CD3. You don't see a whole lot happening to the RAF protein within the first 30 minutes. Whereas if you activate these cells with PMA, 4-ball myristic acetate, and directly activate protein kinase C, then you would get phosphorylation of RAF. And then down here in B, we see the anti-CD3 effect compared with the IL-2 effect. So we don't see too much shift in the electrophoretic mobility of RAF with anti-CD3, but we do with IL-2, and that's accentuated with PMA. Down here is just more of the same here. With IL-2, you see it stimulated starting to go within 5 minutes and 30 minutes, 60 minutes, and then at 6 hours, you can see that there's clearly a shift up and more marked at 14 hours. So IL-2 increases the message, increases the protein, and increases the phosphorylation of the protein. Then in the last figure, figure 4, we look at the RAF-associated autokinase activity. So this is where we immunoprecipitate the RAF protein and add P32 labeled ATP, and we look for phosphorylation of the RAF protein. And you can see here with IL-2, minus IL-2, plus IL-2 at 2.5 minutes, 5 minutes, 15 minutes, 30 minutes, 6 hours, 10.5 hours, 14 hours. And you can see that clearly this is the shift. And you can see that the protein has been markedly altered by the autokinase assay in the autokinase assay. And here's the positive control with 15 minutes of PMA. So that's it. We'll stop the screen sharing on the data aspect of this paper. The discussion of this paper is actually interesting in that a lot of the whole history of the oncogenes and the mouse embryo fibroblasts and so forth are discussed in this instance. And in the very first paragraph of the discussion, we conclude that if you can separate G0 from G1 progression, transition, you can then really do this kind of a study. And when we've done that with T cells, we couldn't do it with fibroblasts. And we find that RAF messenger RNA, protein, and kinase activity all increase progressively through G1 and peak just before the S phase. And of course, that's what we knew from Doreen's experiments, what was happening within the cell system, what we call the IL-2 T cell system. And because of the fact that T cells can exist or do exist physiologically in a state of G0, you can do the studies after the anti-CD3 end of things. And it was noteworthy to us anyway, that the RAF messenger RNA expression in the resting G0 T cells was barely detectable, which meant that if the RAF wasn't expressed in G0 cells, it really couldn't participate in the signaling necessarily of itself, of the RAF messenger RNA. And that had to come about through some other different mechanism. And the other thing that we could see from our experiments is that when you added anti-CD3 to these G0 cells, you didn't get increased in autokinase activity, but you could see that PMA would actually do that. So we wondered, we conjectured and speculated at this particular point in time that maybe there was a PKC, protein kinase C, calcium-dependent protein kinase that was a tyrosine-activated kinase. Maybe there was one mechanism that would activate RAF that was separate and distinct from a different mechanism that was PKC independent. The other thing that we noted is that the progressive increase in the message, the protein, and the kinase activity throughout G1 was what we were looking for, essentially. And we wondered whether or not the IL-2 receptor, because we knew that it had kinase activity, the IL-2 receptor, And it was a tyrosine kinase activity that would activate, would phosphorylate different proteins in the cytoplasm on tyrosines. And we wondered whether or not that activated the RAF, of the whole RAF mechanism. And we ended our discussion by saying that, okay, the next thing we got to do is we need to search for RAF substrates. We need to know what RAF does during this G1 progression phase of the cell cycle. I should say parenthetically that the immunologic community, they didn't care about these experiments necessarily. It was the cell growth people and the cancer oncogene people who were really hot on these kinds of questions. So subsequently, and what happened to us and others in this whole saga is that this was before, just to bring everybody back to where we are in the history of this immunology. This is before JAKs and STATs had been discovered and reported. That didn't happen until 94, 95. And so this is 91. And of course, we did the experiments in 90 and submitted the paper in 1990. Now, this paper that we just discussed came out in May of 1991. In June of 1991, Tada Taniguchi had a paper in Science, no less, saying that the IL-2 receptor beta chain that he had cloned interacted with a tyrosine kinase called LCK, and that's how IL-2 signaling occurred, through the IL-2 receptor beta chain and the tyrosine kinase LCK. And I said to myself, well, that's what the T cell receptor does. How can the T cell receptor and the IL-2 receptor activate the same signaling pathways? Because they do different things to the cell. The T cell receptor cannot activate cell cycle progression, and doesn't, at least in our experiments. Now, the T cell receptor people always sort of felt they didn't want to have to deal with these lymphokines and the stuff that they thought that they were just amplifying this cytokines, interleukin-2, interleukin, whatever. They didn't care. They just wanted to say that the T cell receptor did everything. And I always thought that was, these people are crazy. They don't know. They're not asking the right questions. So subsequently, what happened was, when JAKs and STATs and everything it was a big explosion of all kinds of things, not only IL-2 receptors and so forth, but all kinds of cytokines and cytokine receptors. All the hematopoietic growth factors were involved and used JAKs and STATs. The interferons were involved and used JAKs and STATs. There were 7 different STATs. There were, I think, 4 different JAKs. So it was just a whole cornucopia of new molecules to fiddle around with. And so just what happened to us in this whole, and so that all happened, and that we will get into some of that subsequently in the journal club. What happened in our lab was that because Nina was a graduate student, she graduated, and I elected not to pursue. I was confronted with a situation whereby, you can't do everything, and you have to choose where you put your money in this kind of a situation. And so it looked as though at this particular point in time, it was clear that signal transduction after 1990 was going to be where it was at for an awful lot of people interested in cytokines, their receptors, and so forth. A lot of people involved. Now, it wasn't just interleukin-2 and immunologists. Now we had the hematopoietic factor people, and we had the interferon people, and so forth. So, and a lot of big labs and very accomplished labs in the biochemistry of signal transduction. And I said to myself, this is getting to be a little bit too hot in this area, and I decided to not pursue signal transduction as time went forward. So we didn't pursue this RAF story, but RAF was important, and this paper was important because it was one of the very first papers that showed that IL-2 and IL-2 receptors activated this particular oncogene, and we'd already shown that it activated the expression of CMYB, and other people had shown that it activated CMYC and CFOS and PIM and so forth. So there was a whole building up of data showing that the way you stimulated growth within the different target cells was during these signal transduction pathways and activation. And this was cellular biochemistry. And at this stage of the game, and you can see what the data looked like on those gels and autoradiographs that I showed you. I didn't like the science. I didn't like the assays. The assays were semi-quantitative at best. They were qualitative. It was almost, is it there or is it not there? It's a little bit or a little bit more. And I was really operating under the dictum that Sir Peter Medawar had published in his book, The Advice to a Young Scientist, which he said, you can make a lot of progress in science by quantifying what other people have discovered as a phenomenon, but have left the quantification of that up to others. Well, if you're one of those others, then you quantify what's going on. So I liked assays that were very, very quantitative and not only simply semi-quantitative or qualitative. And for those reasons, because the field was clear that it was going to be hot, and because it wasn't quantitative, I decided to not do that. But rather, what we decided to do, the other aspect of this whole cell growth, biochemistry and MoBio business was the molecular genetics of cell psychoprogression. And as we had already shown, and Julie Stern had shown, that IL-2 would activate the message of CMYB, and in this paper we showed it would activate the message of the transcription of Raf-1. I decided what we really should do was to look for new genes that had not been discovered through the oncogene situation or whatever. And I knew on the basis of experiments that we had done also in collaboration with Art Pardee, because he was interested in exactly the same question that I was, which critical genes are activated during cell cycle progression that actually lead to DNA replication and subsequent mitosis. And so that's what I decided to do. And there'll be more papers to come when we get into that story. So don't forget to like the video, click on the bell, subscribe, and check out the papers on my website. And thanks for listening. It's been fun.